Okay, this video is on the scientific process, also known as the scientific method, or how to plan a scientific investigation. Make sure you are putting this in your table of contents, and when you're done with your notes, you tape or glue it into your notebook. Be doing that with all of your notes throughout the year. At the end of the year, we will be referring back to these also throughout the year. So it is important to be organized. It will pay off later. So the scientific process, this should be somewhat of a review from previous years in science. So this unit is short as to not completely bore you, but to get that needed information and that review. If this is very fuzzy to you, make sure you take the time you need to study before the summative and review things, as this will be popping up throughout the year. Okay, now let's recap our CER notes real quick here. Now, as we go about our day, we can ask questions about things that happen in the natural world. We call these things phenomena. A lot of the times, these questions lead to investigations or experiments. And after we do that experiment, the answer to our question is called the claim, and that is a 1 to 2 sentence statement. We collect data along the way, and we call that our evidence that backs up our claim. And at the end, we want to explain what happened, why it happened. Scientifically, that is called the reasoning. But we don't know everything, so we need to back up our reasoning with credible sources. We need to do research to make sure we are understanding what is happening. So, a credible source, we want to look for a .org or a .edu or a research paper. Okay, now it's important to note here at the beginning that if you do an experiment, you do an investigation, and you get results that are not expected, that is not a failure. You have learned something through that process. A lot of times, students get down on themselves because they got results they didn't think they would get, and they feel like they did something wrong. Well, that is learning in itself, and that is part of science. And the most famous scientists have failed many, many times before getting the right answers. Here's a little quote by Thomas Edison to put that into perspective. I've not failed 700 times. I have succeeded in proving that those 700 ways will not work. When I have eliminated the ways that will not work, I will find the way that will work. And you can apply that to really any area of your life, whether that's sports, hobbies, and your school, academic life as well. Okay, let's get going here with observations. Very important that we are taking observations. They're based on our senses. Most of the time, our observations are based on sight. What do you see? What do you notice? But other senses that you could maybe use in some instances: smell, touch, hearing, and taste. Not very often in biology do we use that sense. Okay, what do I see? What do I notice about the world around me? What questions do I have? Okay, because those observations lead to questions. So, for example, just in everyday life, I know that my mom adds bleach to the laundry and then stains come out of white clothing. That might lead me to some questions if I'm curious about it, such as how does bleach remove stains? Or how does bleach affect colored clothing? I notice she always uses it on white clothing, but what if I were to put bleach on colored clothing? What would happen? Okay. And those observations might lead to an entire experiment to find out the answers. Okay. More on asking questions. It's important in all aspects of your life to question what you see around you. Okay. Not always just believing what you see on social media or online, but looking around and questioning and using critical thinking to find out the answers. Okay, in science, we want to ask testable questions, and those are questions that we can investigate with a scientific experiment. Okay, all of our questions are good questions. We might start out with some very basic Google questions, but as we think more about the topic, that might lead us to some testable questions. Okay, so for example, as I'm thinking about my mom using bleach on clothing, my initial question might be, how does bleach remove stains? Well, that's not a testable question. That is an easy Google question where I could look up the answer very easily and understand it. Um, but a testable question would be, what happens if I add bleach to colored laundry? Okay, I could try it out myself to figure that out hands on. That is a testable question. Okay, hypothesis is a potential answer to the question. Just want to remind you that a hypothesis is not an educated guess. That is something that is often taught in elementary school because at that time in elementary school, you don't have the background knowledge to really create a good hypothesis. So it is kind of like a guess. But here, now that you are in high school, you have the life experience and background knowledge to know that you're not making just a guess. You are giving a potential answer to the question. Okay. So a lot of times we might format that as if we change this, then this is going to happen because blank. Okay. But basically, you want to try to put out an answer to what is going to happen in your experiment. And that's the hypothesis. So, for example, if we change the color of clothing tested, then clothing, colored clothing will turn, red clothing will turn pink in the presence of bleach. Sorry, my face was covering that. All right, investigation. Now, this is our experiment. It's going to test your hypothesis. Now, the parts of the experiment, you do need to know these, okay? The independent variable, or we might just call it the IV, is the part of the experiment that is changed on purpose. The dependent variable is the part of the experiment that is measured to collect data, the DV. A few more important terms. Constants, okay? So if we are changing one thing about our experiment, everything else needs to stay constant. So that is the parts of the experiment that stay the same throughout. Control group, okay? So we have different groups in our experiment, and one group does not get the change. It does not get the independent variable, has no treatment. It is there as comparison. Okay, so if I was testing bleach on clothing, maybe my control group would be water on clothing. Okay, it doesn't get the change, it doesn't get the bleach. Experimental group would be any other groups in the experiment, and they receive different amounts of change. So maybe I'm looking at the amount of bleach where two groups get different amounts of bleach. One is smaller than the other, and my control group is a group that doesn't get bleach, it gets water instead. Okay. Now let's look at an example to help us understand these terms a little more. So let's say our experimental question is, how does the amount of fertilizer affect lettuce growth? The independent variable. So look at this question. What could we change? Could we change the amount of fertilizer, or could we change the lettuce growth? Well, we can change the amount of fertilizer by giving the plants different amounts. Dependent variable. What are you going to measure at the end of this experiment to see if the change affects the experiment. We can measure the lettuce growth in centimeters. What are some constants? You should be able to list quite a few constants, not just a couple, but really list them out. Okay, so the amount of sunlight would need to be constant amongst all lettuce plants. They would need to receive the same amount of water, be under the same temperature. Okay, the way in which you measure them should be the same. The ruler that you use or whatever method you're using for measurement. Okay, the amount of soil or the type of soil. You can continue to list these out and be very specific. There should be many constants. Okay, control group. So if I'm changing the amount of fertilizer, what would my control group be if the control group is to get no change? We would have one plant that will not get any fertilizer, or maybe it's one group of plants that gets no fertilizer. So then what is your experimental group? Okay, we could have one plant receiving a 1/4 teaspoon of fertilizer and one plant receiving 1 teaspoon of fertilizer. Or you could have groups, like a group of plants with a 1/4 teaspoon, a group of plants with 1 teaspoon. Okay, those are your experimental groups. Okay, now we need to collect data, and there's a couple ways we can display our data. All right. It's based on your observations. We'll use tools, senses. So you can use maybe you're using a thermometer, maybe you're using a scale to take mass, maybe you're using a ruler, and you're using your senses. So we'll organize in tables like so. Okay. And then graphs. We'll be talking about graphing briefly. You will be getting a little cheat sheet to help you figure out what type of graph to use. When you get that cheat sheet, make sure that goes in your table of contents and in your notebook. Great. Then we analyze and make conclusions at the end. We want to compare our results to our hypothesis. Okay. Now, here's the terminology we do need to use. Instead of saying I was right, I'm going to say I support my hypothesis if the results match my hypothesis. Okay. There's really no true fact, and the closest we can get to fact in science is a law, like the law of gravity. So we try not to say like this is true and this is not true, or I was right and I was wrong, because a hypothesis is pretty low on the totem pole of fact. Okay, so we go hypothesis, then we go theory. Okay, theory is actually supported by a lot of evidence, more evidence than a hypothesis. Okay, so it goes hypothesis, theory, law. Okay, where law is the closest thing to fact. Okay, so support a hypothesis if it matches. Reject a hypothesis if results do not match. Okay, so we don't say I was wrong. We say I reject my hypothesis. Okay, and then lastly, you use CER to make your conclusions and research a scientific reason for the results so you understand what happened in the lab. Okay, so I hope that was helpful. Have a great day.